Hello, thanks for the invitation to participate in this Congress. I'd like to show you our project we are doing here in Argentina. Our concept is that from the weeds with plant species, we can obtain the bioherbicides to control weeds. This is the point. In our project, we collect plants in different parts of Argentina and we produce plant extracts and we use the extract as bioherbicide. There are prototypes. When we have enough information for each extract, we transfer the prototypes to the industry so that the industry transform the prototypes into commercial product. So what initially or actually is a weed plant species, we transform it in a natural resource, it's a crop, to produce the bioherbicide. There are many forces behind this change. The world is changed to the biologicals. Biologicals in this case are forced to be discovered because population increase and they require better food, better biochemicals to control weeds. There are many weeds resistant to the actual herbicides, and we propose to use a different source like botanical extract to control the plants in the field, the weeds in the field. We have developed a method. The method has two parts. The first part is in the field. So in the field, we collect plants in particular regions, and the plant is separated by organs, leaves, roots, flowers, fruits, whatever. And many times we collect soils, and we prepare the extract using water, just water, and after that we lyophilize the extract so that we have a powder. In the lab, what we do is what we do is a selection of the plant extract so that with different bioassays, purification of the leaves plant extract, and we try to understand the mode of action of the extract, and at the end we transfer the technology to the industry. It is important that the rigorosity of the method so that we can control all the steps we are doing in our lab to obtain a lead. And we have to be sure that at the end of our process, we have a prototype with the ability to kill weeds under field conditions. I'm going to show you 4 prototypes that we have in our company. 3 are bioherbicides and 1 is a fungicide. The bioherbicide, the first one, is a molecule that we named HOO2. We have not much information about this particular bioherbicide prototype, but what we know is that it affects particularly in broadleaves, not narrowleaves. And what you are seeing here is a video where a cutting of a weed is immersed in a bottle with a plant extract in both cases, so that we are checking in this way the movement of the extract through the xylem to the leaves. And when the extract reaches to the leaves, the plant is killed. So that the same effect we see when we spray the extract on weeds that is seen in this part of the pictures. In this case, we learn a little bit more about kaolin and bisnachin that we have isolated from Amibis nigra. In this case, we obtained a paper that were published in a journal of agriculture and food chemistry, and Maria Lucia Travaini obtained a PhD working in our lab. The kaolin and bisnachin the work was done with USDA, so that we, at the end of the work, we obtained a patent with them, and we filed a patent in the August 2021. Kaolin and bisnachin has two effects, like pre-emergent and post-emergent. In this case, we use kaolin, in this case, kaolin, bisnachin, kaolin plus bisnachin, pelargonic acid as a control, and no treatment. We use both together because they are present together in the leaves extract, in the flower extract. We use flowers to obtain the extract. You may say, well, how many kilograms or tons of flour you may have in an extract? Well, in this case, we have 15 tons of flowers when we work with amibisnaga. 15 tons of dry tissue of flowers to produce the extract. And as you see, the effect is better in broad leaves than in narrow leaves, and is better always in the bisnachin than in the kaolin. And the pelargonic acid is used as a control. And we know that pelargonic acid is a post-emergent bioherbicide we use as a control. But in this case, kaolin bisnachin and kaolin plus bisnachin were used also in pre-emergent 40 days after, and you will see that bisnachin is still controlling the weeds in the soil. In this case, we don't know if all the plants or the seeds present in the bisnachin were killed before, so that the germination is inhibited completely. In this case, we are using bisnachin now to be formulated with pelargonic acid, and we improve the ability of bisnachin to cross the leaves barriers, so that at the end of the day, bisnachin works at 2 kilos per hectare. In this particular experiment, pelargonic acid, of course, has no effect as a pre-emergent. This is our best result. This is the extract named 004, H004, and as you see in the videos, we apply the extract on the leaves, and what we observed is a progressive effect. We use progressive because it looks like a systemic effect. Maybe a systemic, but we didn't check it. In this case, we see how slowly the plants are killed with just one application. In the upper part of the picture, you will see the control, no treated plants, and from the upper and the side of the plant when they were treated. It's very, very impressive to see how plants are killed. And in the other cases, kaolin, bisnachin, and 002, plant is not completely killed. We need to apply more than once the extract to kill the plant. In this case, it's very easy to see how the plants are killed. So that we did with this particular extract, again, dry tissue and water is the extract, and twin 20. We did another experiment. Those are the species that are not affected or moderated affected by the extract. Not all the species were affected in the same way. This picture is better to understand the effect. We have here the treated sample, the testing samples. It is the extract compared with the Roundup, and we have the adjuvant. We change it. We use twin 20, we use Dash and MSO. At that concentration is stated here from 40 to 5 kilos per hectare with different grains and weeds. Of course, the glyphosate has a better effect, but we compare among different adjuvants, we see that twin 20 and Dash has a better effect than MSO, and some species are more affected than others. And we change the next experiment. We change the amount, the concentration of Dash from 0.5 to 1%, and we increase the amount of the extract from 60 to 7.5 kilos per hectare. It's curious for us that in broader leaves we have effect at a low concentration, as low as 7.5 kilos per hectare. What it means is what the chemist said, that we can improve the concentration, diminish the concentration between 50 to 100 times, so that we can use a lower, if you have a pure compound, we can use a really very low concentration of the extract to produce the effect that we are seeing here in the broadleaf, if that is the case. The extract has ability to work with some weed, not with the others. The adjuvant, the better was 2020 dash, and the efficacy is different. It depends on the weed we are working with, so that at the end we have to purify the molecule that is producing the ability to the extract to be transformed in a bioherbicide. We don't know yet which is this particular molecule, but we are working with a company to purify this active compound. This is a different scenario. We are now working with a fungicide. This particular plant is potato. We spray an extract on potato plants, and 24 hours later of the spray, we infected with Phytophthora. What you see here on the left is that the plant was affected because it has no treatment. But in the other two cases, the extract was applied at two different concentrations, 5 kilograms per hectare, 2.5 kilograms per hectare. And in both cases, the extract was good. What is interesting for us is this was really interesting, but more interesting was that the extract did not kill the fungus directly. There is not a direct effect of the extract with the fungus. So we did a different experiment. In this case, we are showing in this slide, we have a plant not treated, a plant sprayed with nothing. In this case, sprayed with water, and the other one is sprayed with water plus 20. And in this case, E3, the plant was sprayed with the extract. And you see how the first and the last pictures show that they looks like the same, so that there is a protection. In here below, we use Petri dish. What we did here, it was really interesting, because we cleaned the leaves, we detached the leaves, we apply the extract. After a few hours, we detach the leaves, we wash the leaves with water, and we put the leaves on the Petri dish. At the end, they were infected, and the effect was triggered inside the leaves. So what we think is that the plant is producing an effect like an elicitor effect. We did RNA seq, and we determined that many genes were expressed during the application of the extract. We use also an experiment in the field, and we compare in potatoes plants too, and we found that it's very interesting when we compare with commercial product in potato in a farm, so a region where people is producing potatoes. And here on the right side, we are using pepper, and it was a production in Chile. They produce peppers, and it was, in this case, it was a particular, because we applied the extract not as a pre-infection, but also as a post-infection. The extract, in this case, cured the plant produced fruits, and the fruits produced seeds, and the seeds were not infected by the fungus. It is great because Chile produced 60% of the seed from this particular crop worldwide. So that for them, the production of seed is very important, and many times seeds contain the Phytophthora fungus, and is the way that plants or the crops are contaminated. So we did this experiment using a different method to dry the plant tissue, so that we use a spray and determine the conditions, and it can produce a lot of kilos if we want to. And in this case, the antifungal effect was not observed or was very weak, but what we have in our hands is an elicitor. It's a very, very good elicitor to control the Phytophthora infection. In our lab, we are Maria Lucia Travaini. We started the project together. Lara is a PhD student, was incorporation later. Tomás is now the PhD student, and Imanol is our technician. Eduardo and Nestor are our consultant from the National Council of Research. Dr. Stephen Duke, I think everyone knows Stephen Duke. He's a professor in Mississippi University and a researcher in the USDA. Dr. Helmut Walter is retired from BASF, from the research and development of agrochemicals. Littoral is an accelerator program. They bought part of INBIOAR a few years ago. Federico and Sebastian are both investors on the INBIOAR, and Lichi, Lisandro Rosenthal, is our family investor, family office investor in our project. A last word, what we are doing. What we are doing is collect plants in the field, produce plant extracts, lyophilize the extract, and apply in different widths, broad leaves, narrow leaves. And at the end, what we are doing is proposing a different way to produce biologicals, in this case, bioherbicide. Of course, we do insecticide, fungicide, and also stimulants with the same plant extract. We have the extract there, we have the ability to do biotest. So it's a way to have very, very important transformation of the weed plant species into a crop. And we propose to control the weeds or the pest in general terms with a biological tool. I hope in the future to repeat our project in Chile, Paraguay, United States, India, and Africa and Australia. Thank you very much for your attention. It's my pleasure to be here with all of you, and I am open to your questions. Thank you.